Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Hilary Eaton, a rare disease advocate and chief business officer at Profluent, an AI-first protein design company on a mission to accelerate biomedicine. Welcome, Hilary.
Thank you so much for having me, Nicole.
So today we'll be discussing how AI and LLMs are changing the drug development paradigm and its limitations. But before we sink our teeth into our central topic, Hilary, perhaps you could share with listeners a little about yourself and the passions and advocacy that led you on your journey to doing what you do today at Profluent.
Happy to. I always am intrigued by that question that you get asked of how do you know what you want to be when you grow up, and how that can sometimes change quite dramatically and evolve as you, develop in your career. I started out with a PhD and a postdoc, so working at the bench doing cancer research. And at some point in that process, realized that a lot of the topics that were being worked on in academic research were very niche areas, mechanisms, how do things work? And there was a disconnect, I think, between some of that very foundational research and how do we turn this into a medicine that's actually going to help a patient. And so thought that maybe I'd like to get a little closer to that patient world. And so segued into the business side of science, moving through a few years at the Tech Transfer Institute at Inner which is focused on trying to figure out how to turn some of those basic research innovations into companies or licensing to big pharma, and then into a series of business development roles at gene editing companies that use CRISPR-Cas9 to address genetic problems. So Editas, Vor, and Tome. And, I thought of CRISPR, it sounds so sci-fi, this idea that there is now a tool that can go in and fix something that is wrong in somebody's DNA. So not a pill that alleviates symptoms, not something that's sort of like tangential to, uh, but really sort of just going in, finding what's wrong in your DNA and fixing it. Um, so that really captivated me. And whether sort of like ironically or serendipitously, I don't know how to characterize it, but those early years for me at Editas and Vor, a lot of the work is spent thinking which patients can we help with these technologies, and looking through reams of genetic diseases and their profiles in terms of, is the tool that I have a match to fix this thing? And everybody's in it for the patients. That is central to the mission of any drug development company. And yet there's also a little bit of that you feel empathy and sympathy, but it's other. Like you're reading through these patient profiles of other families, other kids, other moms, and these horrible things that happen to them. And as I progressed throughout my career, here comes the irony. It turns out that myself and my two young daughters have a rare genetic disease that I was completely unaware of at the start of this career journey. And so then that takes an issue that could be perceived as being a little bit more abstract, hypothetical arm's length, and takes it to, no, this is my kids. What wouldn't I do for my kids? And so that's kind of led me to where I am today as Chief Business Officer at Profluent. It's a unique combination of my passion for rare disease patient advocacy and the growing understanding that there are many ways that AI can help in drug development, not just in parsing data down the line in clinical trials or helping with CMC or QC, but really at the drug discovery and development stage.
Thank you, Hilary, and thank you also for sharing your personal journey into rare disease patient advocacy. So if we bring it back to the technology now, let's dive into AI and LLMs, as you say, in drug development, not just parsing data. So briefly, if you could, can you run through where we've come from to where we're actually at now and what might just very well be right around the corner?
Happy to. So let's take a tool that hopefully a large portion of your audience might be aware of. So AlphaFold 3. I would characterize that as essentially a reader of biology. So you provide it with the sequence of a protein, and AlphaFold 3 reads it and predicts how it will fold, what its structure will be, and equates that to a functional outcome, right? And that is an amazing advance that lots of people are using for some really exciting drug development. Um, what Ali, our founder and CEO, set out to create is a model that can write biology, not just read it. So if you draw parallels over to the world of natural language processing and think more about how has have things like ChatGPT changed the world, if you were to, rewind in time 5 years, 8 years, often as you're sort of typing on your early generation iPhone, uh, autofill couldn't even finish a word, let alone predict what the rest of the sentence is going to be. And as researchers at places like OpenAI and all of the big tech developed new ways of building these foundational models, and with the advent of things like the transformer algorithm, you got these really powerful models that were trained on millions examples of text and could play all of these simple little games over and over again in parallel so that they learned things like fill in the blank and what do different styles of writing look like and what's correct grammar and syntax and all of that fun stuff. And that has culminated now in tools that millions of subscribers use as part of their everyday life, which didn't exist 2 years ago. Models that can, take any series of prompts and provide you with coherent text, accurate facts, in whatever style you've asked them to do so. And so then how do we translate that to the world of biology? And so when Ali was at Salesforce, that was really one of the central questions he had is that if you look at a protein sequence, it resembles a string of characters, which resembles English language. It just codes for a protein, and most people can't really read that series of amino acids. And so he thought, could we take some of these large language models and instead of exposing them to examples of written English text, could we expose them to millions of protein sequences? Would they be able to learn the underlying language of life? And, somewhat to his surprise, they could. So in early research that he published, it showed, an ability to take on um, smaller, simpler enzymes. And, for you to treat the model sort of, to give it a prompt almost like you do with ChatGPT to say, I would like to build this type of protein that has these criteria that will solve this specific problem. Can you write it for me? And to see that the model indeed was able to do exactly that in a zero shot manner, which means that this was just based off of what it had learned from being exposed to this large array of natural protein sequences. There was no wet lab data involved or iteration, et cetera, et cetera. And so that, that kind of was where we were a few years ago. Fast forward to now, and these models are able to do it with much more complex proteins on many more parameters and even think about introducing sort of new to nature functions. So, exploring avenues that perhaps evolution never got a chance to explore and creating bespoke things that are intended for very specific problems. So rather than sort of look around the natural world and say, can I find something accidental that might be a fit to the current therapeutic or biomanufacturing or climate problem I'm trying to solve? Can I instead intentionally start with the problem, brainstorm exactly what the solution would need to be, and then ask my model to build it?
As you say, Hilary, it all sounds so sci-fi. It's fascinating and nearly unbelievable to the extent at which it has been stretched and indeed achieved. So if we turn now to gene editing, last year, researchers at Profluent used artificial intelligence to design novel CRISPR gene editors that are functional and that show comparable or improved genome editing activity and specificity relative to naturally occurring gene editors, as reported by CRISPR Medicine News, despite being hundreds of mutations away from any known natural protein, as you were saying, is this ability to explore what evolution itself hasn't yet had the opportunity to do so. So this innovative approach from Profluent yielded OpenCRISPR-1, the first AI-generated gene editor, I believe. So I was wondering if you could tell us more about this.
Absolutely. So, going back to sort of like the sci-fi or the hypothetical nature of, AI and its role in drug development, it's fantastic to say, I have the best models, I have the biggest models, I can do all of, what you do with it, that is the crux of the matter. And so, as Profluent was considering launching and coming out of stealth, there was the thought that we would do a proof of concept with our models that A would demonstrate the type of problem that necessitates or most behooves using AI to solve, i.e., not a smaller, simple protein, but something that is incredibly intricate and complex. And Cas9 certainly fits the bill for that. Um, and secondly, for this POC, to design something that could be useful, not to create something just for, the whim of I can create this thing, but to create something that we felt that researchers might be able to really use and integrate into their research. And so the idea came up to look at Cas9, see if we could create a copycat or an analog, something that functions very similarly and yet looks quite different in terms of its sequence identity. And this POC didn't really even showcase the potential of our model in terms of sequence novelty because we constrained the model to be able to do an accurate apples to apples bake off. We wanted this gene editor to recognize the same sequence that Cas9 did, so having the same PAM and accept the same guide RNA. So there were these constraints placed so that we could create something that you really could compare side by side to Cas9. And so did this and made the somewhat unique, actually very unique decision to open source. So that is very much a tech paradigm, right? There's a lot of sort of open sourcing that you see in tech and software in models. That's not something that you see very frequently on the medical side. There's a lot of sort of hoarding of knowledge or very carefully protected IP landscapes because that's the thing that you're going to build the business on. For us, however, this was just one proof of concept and generated millions of molecules, all of which had pros and cons and various different features. And so we thought, why not make it available to see who might be interested? And I was absolutely shocked. Like normally on the BD side of medicine, when you have something available for licensing that's going to be used as a human therapeutic, you're lucky if you have one bidder, two bidders, maybe three bidders for a given technology. I actually just checked our Open CRISPR license request form, and we have passed 1,100 license requests for this free molecule. That is just unprecedented volume. And it's fascinating to see what people are doing with it. You have folks from agriculture trying to create more sustainable crops. You have folks on the biomanufacturing side. You have folks on the human therapeutics side. And ultimately, it came down to a lot of them have amazing ideas for how they'd like to apply gene editing to their specific area of technology. And yet, some of the other enzymes that exist in the space that are more clinically de-risked, the IP landscape is very murky as to who owns what rights in which geographies. And the royalty stacks, the amount that they would have to pay to the original inventors, are so high that it almost becomes prohibitive of them actually developing anything with those enzymes. And so for us, it was a great proof point of technology being able to create something that has, immediate direct avenues of use by folks across a wide variety of technologies. We were just really happy to see sort of the molecule going out and being test run in so many different scenarios.
Well, I think congratulations are in order, firstly. Um, but my next question is about ProGen 3. So, Profluent has also recently announced a new frontier model suite, which is ProGen 3, and it demonstrates with real world evidence, and correct me if I'm wrong, that AI scaling laws, which predict model performance based on size and computing power, apply to biology. So tell us more about this concept, Hilary. But I think you've been leading us along, listeners along to this question about this AI applicability to biology to the point of preemption of evolution.
Absolutely. And so, again, if you hearken back to the world of natural language processing and more the GPT world, there was a lot of research during the creation of GPTs 1, 2, 3, 4, etc., that was exploring the relationship between the amount of data that was being fed to a model, and then the size of the model itself. And the idea being, if those are the axes, amount of data, size of the model, what did you see in terms of model performance? Did it correlate with being exposed to more data and with being bigger, or sort of, you know what I mean? And it was a very linear relationship that as you increase the amount of data, as you increase the size of the model, the performance of your model improves and improves and improves, which is obviously something too that like investors are very interested in, right? Why pay more money for gathering and curating more data? Why pay more money for building better models unless you're actually improving the deliverables that you get out of these models? And so that is something that really has not been demonstrated for biology itself. It's been demonstrated for written English text. And so we became quite interested in this, phenomenon of could we replicate some of that on the biology side? So in this case, it's number of protein sequences that a model is exposed to in its pre-training and the size of the model that you're building. And so we came out with this preprint where we have similar model architectures, but built in sort of like a conservative small size, a base size, and an extra large size. And these were trained on an extraordinary number of protein sequences. And then we looked at how those models performed in terms of did the number of hits that came out of a protein design campaign increase, and were the confidence scores higher? And in fact, they did. So the extra large version was much better than the other two at meaningful functional outcomes in terms of what it was able to provide. But it goes a little bit further than that. It's not just does your general model performance increase? Are you getting, a higher percentage of successful hits? We also saw some evidence of emergent phenomena. And stay with me here because I'm also not an AI expert. I'm learning this on the fly. But essentially, to compare it to, again, the language world, there are certain things that a little kid just can't do with language, right? If you ever watched a small child learning how to communicate, and there are some things that they just cannot do. They haven't had enough real world experience. They can't make these intuitive leaps. And then as they grow up, all of a sudden, without necessarily a lot of active, proactive training, they've been exposed to more things. They've learned, somewhat passively how to do these things. And bam, you have a fully functional 10-year-old that's talking back at you, right? And that's exactly what these models are doing, is that you're seeing the largest version suddenly being able to create things in its protein that it's writing that you would not have expected it would have been able to do. It's making sort of these leaps, and all of a sudden complex functions that you might have asked earlier or smaller versions of the model to make, it couldn't. And now all of a sudden it can. Right. And so that's really exciting because then you can start thinking, a little bit broader than, say, I want to do this. We get all sorts of partners coming in and saying, I would like this protein to be more temperature stable. I want a better version of this. You can now really start thinking outside the box into not just what would be the incremental improvement to this protein. But if I could just imagine the thing that would be wildly successful for this particular problem I'm trying to solve, let's task the model to do that thing.
As you say, excitement indeed. So, if we stick with this excitement over emergent phenomena, emergent potential, and the ability to now start thinking outside of the box, when it comes to the acceleration of biomedicine, what are your takeaway thoughts and hopes for the space in the next, let's just say 5 rather than 10 years, Hilary?
I mean, I'm almost hesitant to make predictions because it feels like it's moving so quickly. You know what I mean? How are you supposed to tell where it's going to be in 5 years? I am, coming from a history of wet lab and sort of traditional molecular biology training. It blows my mind every day what the team at Profluent can do. And that's actually a really key point that I'd like to stress, is that I think some of the hype around AI centers around it's, for lack of a better word, a magic wand that's just going to fix problems. And it's not that. It is a incredibly powerful tool that when used by the right team can be used to solve problems, right? you have to teach the model things. You have to guide it to do certain things. It's not just creating proteins by itself. You're the one that's going in, a team that's got deep expertise in protein structure, in whatever the therapeutic or other vertical is, biomanufacturing, agriculture, manufacturing, and saying, from what we know from our experience in these areas, what would be the thing that we would tell the model to make? What should it avoid because that's something that's less manufacturable? Do we really care about immunogenicity? What are the hot topics in those areas, right? And guiding it together. And that's when you really start seeing the magic happening. Um, and you need to be really safe and ethical about it, right? I I think that more and more pharma is coming to the point where they recognize that this is a huge tipping point or inflection point because everybody is starting to incorporate it. And to various degrees, right? You'll see some companies where they kind of sprinkle a little AI on top to sort of like get in and on a little buzz. And then you'll see companies that are innovating on the machine learning side. So they're not only pushing forward programs that are drugs designed by AI, but they're also making innovations on the machine learning and the model front themselves. And I think, what's going to be really interesting over the next 5 years is to see how those approaches sort of pan out. If you're trying to bolt on AI, but you're not exactly sure how to use it or how to implement it over a very large company, versus if you are born with AI as part of your bone marrow and it's with you from day one, what's that going to look like? What types of collaborations are you going to see between small and large? Who's going to scale, I guess, on the AI side? And again, it comes down to use cases, right? Take your sort of gnarliest problems in human medicine and start imagining, you know, what would solve that problem. And I don't think that anybody would claim that anybody's AI model is going to, just left, right, first shot, be able to solve a problem in one go. But again, it's an immensely powerful tool for going after some of those most difficult problems, where it's just not manually possible for a person or a team of people to search through all the possible combinations of amino acids that could make a protein that could solve that thing.
I like that expression, applying AI to the gnarliest problems in human medicine. If that's the takeaway, there you go. Thank you so much, Hilary. It's been an absolute pleasure.
Likewise. Thank you so much, Nicole.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.